Here we go with chapter 19. It seems really hard. We're talking about eukaryotic genomes. Now, just to review here, the genome is all of the genes, DNA in an organism. Okay, so all of the genes and all of the DNA in an organism. And so we're just really talking about, okay, how in the world do genes turn on and off? And then what are the results of certain genes turning on and off? Okay, first of all, organization. There's a few slides here talking about how 6 feet of DNA can fit inside of every cell. And it's packed. DNA is wrapped around proteins called histones. Histones, right here, you can see that it makes DNA look like beads on a string. The double helix of DNA is wrapped around these things called histones. It is thought that unpacking of DNA is the first step in making a protein. So you have to unwrap the DNA in a spot to make a protein. Okay? So anyway, if we go on, then those histones are packed together here. And you see a whole bunch of histones all packed together. You don't have to remember the name nucleosome. And then they have what's called a protein scaffold. They're looped up and down around a structure. And then those loops are folded again and wrapped up to make a chromosome. And this is a chromosome at metaphase with 2 copies of the DNA. And so you see that the DNA is all very much coiled and wrapped, and it fits inside of your cells just fine, even though there's 6 feet of it. This slide is an overview of what we call genetic control, or how we control the processing of genes. Okay, in other words, can a gene be always on and not express itself? Yes. Can a gene be always off and be expressed? Yes. Okay, you get some kind of signal to the cell. Okay, some cells, like in your pancreas, there has to be a signal to turn on insulin, turn on the gene to make insulin. Okay, and then you have to unpack the DNA. You have to open up the gene, make the gene available. Then you have transcription. Okay, where, remember, something like A, C, G, T, U pairs with A, G pairs with C, C pairs with G, and A pairs with T. Now you have what's called the primary transcript. Here's the first set of processing, is that you have to remove the introns. So you see that this piece of RNA is longer than that piece of RNA. Then a cap and a tail is added to help it get out of the nucleus. So here's our messenger RNA now with just the exons and a cap and a tail. Now sometimes this all happens and messenger RNA is not needed. So then it's degraded or gotten rid of. It gets out in the cytoplasm. Well, we've made enough insulin. We're not going to produce any more. If it is needed, it goes into translation. Translation is at the ribosome, and a polypeptide is made. Okay, a polypeptide, remember, is not a protein yet. It's a primary, it's the primary structure. It's all the amino acids all chained up. It still has to be converted into an active protein. Sometimes, though, the polypeptide is made and the protein is not needed anymore. Then it can be cleaved or split apart. It can be chemically modified, or it can be transported. Or it's made into an active protein by the Golgi apparatus. And now you have the active protein, but what if you don't need it anymore? What if you got all the way through and made enough insulin? Well, then these really sweet things called proteasomes that look like little trash cans get rid of the extra protein. break it back up into its individual amino acids. So those are the steps in genetic control, if you will, or controlling how genes are made. Another method of gene control is called DNA methylation, where methyl groups are added to certain bases in DNA. Sometimes it inactivates genes for a long time, okay, or maybe ever. And so it's thought that maybe in our forehead, for example, the gene for hair is methylated, okay, in our forehead skin cells. In genomic imprinting, methylation turns off. That's how it's done, to turn off. We talked about that briefly when we talked about genomic imprinting. Okay, this is an overview of transcription to RNA processing again. Okay, except we need to talk about what turns on genes. How is it possible that we can turn on genes? And we really don't know. But we have an idea that, okay, this is the DNA and here is the gene. Okay, here is the gene. There's transcription. We have exons and introns. Okay, we get rid of the introns and we have the exons. Add the cap and the tail down here. Upstream, towards the 5' end of the DNA, is what they call enhancers and control elements. Confusing terms, but things they think that are involved in turning on genes, just like in an operon in bacteria. And so you're going to read about the term transcription factor, okay? And a transcription factor is a protein that has to be present in order for a DNA to be read. Okay, and then there's other things, mediator proteins and DNA bending proteins, and that's not that important really, except that RNA polymerase has to go along the DNA and create RNA. And in order for that to happen, it has to start at a spot, and it always starts at something called the Tata box, where it's a series of TATA upstream from the gene. The gene is this way, and that's called the promoter region, just like in an operon. Transcription factors are needed in order for this to attach and go. Just like a promoter or an operator is needed, an on switch. That's kind of like the on switch to go through the whole process. So, and there are some transcription factors that are actually repressors to inhibit expression of a gene. So in human genetics, we have activators and repressors. And we actually know most about these from the genetics of cancer, actually. Here's an example. We're looking in two different cells. The liver cell nucleus. Liver has to produce a gene called albumin. Okay? And so we have available activators or transcription factors in the liver cell that stimulate production of the gene. In the lens cell, we have to produce a gene called crystallin, which makes sense. Okay, in the lens cell, the activators are different than in the liver cell. So the albumin gene is not expressed because it doesn't have the right activators, but the crystallin gene is expressed because it has the right activators. Now, this is very theoretical. Okay, we have a little bit of proof of this, but not really that much. As we talked about before, here's a view of a proteasome. The protein has been made. Okay, we've gone through transcription and translation. Protein is made. Here's a protein, and there's something called ubiquitin. Ubiquitous means everywhere. A protein called ubiquitin that gets attached to this other protein that you made. Here you see the tertiary structure of it, or quaternary, I'm sorry. And there's proteasomes, these giant trash can-like things that break up proteins that aren't needed back into their amino acids. I just think that's really cool. So what about cancer? Well, researching cancer has really helped us to understand this, how genes are controlled. And so let's talk about briefly about the genetics of cancer. Let me move this a second. There are genes that normally regulate cell growth and division during the cell cycle include genes for growth factors. In other words, genes that make the cell grow. They're receptors, the proteins in the surface of the cell that pick up signals, and then other signaling molecules. Any mutation in any of those genes can lead to cancer. A couple examples. There are genes called oncogenes that we know cause cancer. There are genes called protooncogenes, which are normal genes that can be converted to an oncogene, usually having to do with promoting excessive cell division. And so, yeah, I don't think you really need to know this, but here's the normal growth protein. Okay, so a proto-oncogene can have problems in many ways. We can get too many copies of the gene. We can get mutation in control element to make it produce too much growth protein, or we can get a mutation in the gene that makes a hyperactive protein. There are also tumor suppressor genes. Okay, and we're going to talk about both of those here in a second. So we're going to look at two genes, a proto-oncogene called the RAS gene and a p53 tumor suppressor gene. All right, the RAS gene is a cell cycle stimulator. Okay, here comes the growth factor telling the cell to divide. Divide, cell, divide. Here's the pathway. We have the receptor, we have the transduction, and we have the response to produce a protein that stimulates a cell cycle, a kinase or a cyclin-dependent kinase or something like that. Okay? So the RAS protein is hyperactive for some reason. It starts issuing its own signals. So you don't need this for this to happen. You don't need the growth factor for stimulation of cell cycle when you normally would, because this protein is issuing signals on its own. That's bad news. The p53 gene is a tumor suppressor gene. Okay, and it's called the guardian angel of the genome. It's involved in stopping the cell cycle. So if you have this happening, that you're making too much of the cells, it'll stop it, unless it's mutated. Now let's look at this whole thing. Okay, here's the one that I just showed you with the RAS protein. Here's an example of a cell cycle inhibiting path where for some reason, let's say you've been out in the sun too long and you damaged your DNA. Enzyme or protein kinases are made that activate p53. p53 is a transcription factor. So it attaches to the DNA and makes it produce a protein that stops the cell cycle. Remember, we just talked about transcription factors. But let's say we mutate that gene. We mutate it, knock it out. Well, if we knock it out, now we don't inhibit the cell cycle, so we keep making damaged DNA. Every cell that divides makes a new damaged DNA, and eventually we have cells that are all mutated. So what we really have in cancer is a combined effect. You get the hyperactive RAS protein making the cell stimulate the cell cycle too much, and a mutated p53 gene. Usually it's many mutations. More than one somatic mutation is generally needed to produce a full-fledged cancer cell. About a half a dozen DNA changes must occur for a cell to become fully cancerous. These cells usually include at least one active oncogene and the mutation or loss of several tumor suppressor genes. Here's an example using colorectal cancer, okay, where you get, here are the normal cells, you lose one of the suppressor genes, and you get this what is called a benign growth. Well, sometime in there, the RAS you get into cell division like crazy in your colon because you're always losing cells from there. So you lose another tumor suppressor gene, the RAS oncogene is activated, and it keeps growing. And then you get a mutation of the tumor suppressor gene and maybe some other ones, and it's enabled to spread. It's the general basis or general theory of cancer. Okay, there are certain viruses. We talked about this, maybe not in this class. HPV is a human papilloma virus. It's present in 95% of all cervical cancer cases, cervix in a female. Well, because they put their viral DNA into the cell's genome, and then maybe it just so happens to plop it right into one of these other genes. They think that about 15% of cancers are caused by viruses. Okay, and so then if you think about it, it is possible to inherit mutant oncogenes or tumor suppressor genes. Okay, it's possible to inherit that then. So the important thing to know here is that most eukaryotic genomes have junk DNA in them. Okay, we're stepping aside from cancer a minute. This slide shows DNA, the amount of DNA, what it is. There is, and most of this you don't have to know, unique non-coding DNA. DNA that means nothing but is unique to you, 15%. Introns and regulatory sequences, control elements and stuff like that, 24%. Repetitive DNA that includes transposable elements. This means elements that can be flipped around, moved from place to place, but don't really do anything else, 44%. Total repetitive DNA, 44 plus 15 is about 59% of your DNA is repetitive. And exons, part that's encoding for something, 1.5%. So where did all the rest of that come from? Years and years and years and years and years and years of DNA of organism development. A brief thing, this lady's name is Barbara McClintock. She identified the fact that genes can move around within a genome. In other words, something on chromosome 1 that might be in one spot can move to another spot on chromosome 1. Okay? So, gene that was over here on chromosome 1 can move to a different spot. Well, why would that be significant? Well, if you have a chromosome here and the gene the gene is moved when crossing over occurs, that makes a lot of different changes. And crossing over is one of the big reasons for changes in your genome. Okay, and they're called transposons. They can move from one place to another. They used the term before, jumping genes. They can jump around in the DNA. Okay, there are things called multi-gene families. The globin genes. Hemoglobin is made of two different chains of protein, alpha globin and beta globin, and they're located and they look very similar. They have essentially the same DNA sequence. But you see they're located on two different chromosomes. And to make that whole protein, you have all of these in a row. You don't have to know all this, but this is produced in an embryo, this is produced in a fetus and adult, and these are produced along the way. And the beta globin gene family, same. So if you just need to really know the term gene family there. So this whole slide you don't need to know really. Okay, but the idea is that duplications, rearrangements, and DNA mutations contribute to genome evolution. In other words, the changes in the genome over time. Okay, changes in the genome over time. And we can talk about that in some gory detail in the next unit.